Hello and welcome back. Today we have brought for you part 2 of JNU 2020 MSc Life Science entrance exam paper with verified explanations that you can trust. So let the fun begin. Our first question is, given below are two statements. One is labeled as assertion A and the other is labeled as reason R. In the light of the below statements, choose the correct answer from the given options. Assertion A says DNA vaccine utilizes plasmid DNA encoding antigenic proteins that are injected directly into the muscle of recipient. And the reason is DNA is taken up by dendritic cells and is able to induce both cell-mediated and humoral immune response. So yes, DNA vaccines work by injecting genetically engineered plasmid containing the DNA sequence encoding the antigens, against which an immune response is sought. So the cells directly produce the antigen, thus causing a protective immunological response. The DNA plasmid carrying the antigens are injected into the muscle. And the dendritic cells, or the DCs, play a critical role in induction of immune response by DNA vaccination. Since dendritic cells are antigen-presenting cells of immune system, once activated, they might migrate to the lymph nodes where they interact with the T and B cells to initiate and shape the adaptive immune response. So the correct answer is both A and B are true, and R is the correct explanation of A. Our option number A is the correct answer. The next question is, a diploid eukaryotic cell possessing 3.2 into 10 to the power 6 base pairs will have how many nucleosomes? So in a diploid human cell, for every 200 base pairs, one nucleosome is present. Therefore, the number of nucleosomes in a diploid cell contain 3.2 into 10 to the power 6 base pairs would be 3.2 into 10 to the power 6 upon 200. That is 1.6 into 10 to the power 4 nucleosomes, and that will be our option number B. The next question is, which of the following statements about the complement fixation are true? So let us read out the statements and see which one of these are correct. C3 convertase is in alternative pathway is C3b, BB. So as you can see here, the C3 convertase is C3b, BB. So statement number A is correct. Our next statement is C3 convertase in classical pathway is C4b2A, which is also correct. Yes, we can see that C3 convertase in the classical pathway is C4b2A. Our next statement, statement C, is both lectin pathway and alternative pathway are initiated by binding to C1q. No, actually the classical pathway is initiated by C1q, so this statement is incorrect. Next, C1s has substrate C4 on only. As you can see in the figure, C1s also acts on C2. So this statement is also incorrect. The next statement is C1q and C1s bind to Fc region of immune complex and induce conformation changes in C1r. However, C1q interacts with the Fc site antibodies, so this particular statement is also incorrect. Hence, the correct answer to this question is option number B, A and B only. Moving on to our next question, which is a bacteriophage of genome 3000 base pairs was found to code for 5 unique polypeptides, 500, 300, 200, 150 and 100 amino acids. This would suggest that. So we know that the genes that code for protein comprise of open reading frames, or the ORFs, consisting of a series of codons that specify the amino acid sequence of a protein that that particular gene codes for. And a 3000 base pair phage genome to code for peptides, which are 500, 300, 200, 150, and 100 amino acids, the fudge DNA must have overlapping ORFs. The correct answer to this question is option number A. Our next question is, during inflammation, when white blood cells from blood stream travel into the surrounding tissue, leukocyte extravasation is the central feature of cell trafficking and involves the following events. Now, let's read out the events: activation of chemokines, transendothelial migration, tethering and rolling mediated by selectins, and arrest and adhesion mediated by integrins. We have to arrange all these steps into the correct sequence. Now, here are the steps of leukocyte migration through the endothelium. The first step is capture and rolling, which is mediated by selectins. The second step is activation, in which chemokines and other chemoattractants induce the intracellular signaling. The third is arrest, which is mediated by activated integrins and their endothelial Ig superfamily counter ligands. And finally, the transmigration happens. So as you can see from here, the correct answer would be option number D, C, A, D, and B. Our next question is, an electric field is applied to a semiconductor and there are n number of charge carriers with the average drift speed of V. Which of the following option is true if temperature is increased? So when the temperature is increased, then more charge carriers are produced due to breaking of covalent bonds. And the average drift speed, which is V here, decreases with temperature due to increase in the V and the decrease in relaxation time. So the correct answer to this question would be, according to this particular equation, as the temperature increases, N will increase and V will decrease. So option number D is the correct answer. Next, given below are two statements. Statement 1, etiolated seedlings of corn show absence of greening, reduction in leaf width, failure of leaves to unroll, and elongation of polyoctile and mesocotyle. Statement 2 is etiolated seedlings of mustard show absence of greening, reduced leaf size, hypocotyle elongation, and maintenance of apical hook. In the light of the above statement, choose the most appropriate answer for the options given below. The correct answer to this question is option number A. Both statement 1 and 2 are correct. Etiolated plant is a plant which has been raised in insufficient light or even under total darkness. Etiolated plants, they have whitish or yellowish coloration, which is due to the absence of chlorophyll. Their stems become greatly elongated. There's a poor development of leaves, mechanical tissue, and stomata. Our next question is, match the type of water movement in list 1 with their corresponding mode of transport in list 2. In apoplastic movement, water moves from cell to cell through the intercellular space or wall of the cell. Symplast refers to continuous arrangement of protoplast of plant which are interconnected by plasmodesmata. So, symplastic movements occur from protoplast to protoplast via the plasmodesmata. The transcellular transport occurs through the epithelial cells, passing sequentially through the transmembrane proteins in the apical and basal plasma membrane and the intervening cytoplasm. It is the prominent route for most transepithelial transport. Further, guttation is when a plant pushes water through tips of the leaves of the plant. The process of guttation occurs when the root of the plant absorbs water from the soil. This creates a pressure inside the plant and the water is forced out through the hydrotodes. So the correct answer to this question is going to be option number C, which says A matches with 2, B matches with 1, C matches with 4, and D matches with 3. Our next question is, hemoglobin binds to oxygen molecule via its. The part of hemoglobin molecule that binds to oxygen is called the heme group. The heme group is a prosthetic group in the hemoglobin protein. The heme group consists of an organic compound and the central iron atoms. So the organic components are called protoporphyrins and is made up of 4 porphyrin rings. The iron atom lies in the center of the protoporphyrin bonded to the 4 pyrrole nitrogen atoms. Under normal condition, iron is present in the ferrous, which is Fe2 positive oxidation state. And the oxygen binds to this particular iron ion, which is present in the porphyrin ring. So the correct answer to this question is option number D, Fe2 positive ion in the porphyrin ring. The next question is, given below are 2 statements. Statement 1, RFLP is used in DNA fingerprinting, and statement 2, RFLP is a codominant marker. In the light of the above 2 statements, choose the most appropriate answer for the options given below. Because DNA is unique to an individual, we can use DNA fingerprinting to match genetic information with the person it came from. The restriction fragment length polymorphism technique cuts out the gene which are likely to be differentiating factors using restriction enzymes. The restriction fragment length polymorphism, or the RFLP technique, exploits variation in DNA sequence. DNA from differing source will have variation or polymorphism throughout the sequence. The RFLP as a molecular marker is specific to a single clone restriction enzyme combination. Now, most RFLP markers are codominant. Both alleles are in heterozygous samples will be detected, and highly locus specific. Short, single, or low copy number genomic DNA or cDNA clones can typically be used as RFLP probes. So, both statement 1 and 2 are correct, and our option number A is the right answer. The next question is, match the following organisms in list 1 with the corresponding disease they cause in list 2. Okay, the majority of urinary tract infections are caused by the bacterium Escherichia coli, which is found in our digestive system. Second, the sore throat or strep throat is an infection in the throat and the tonsils caused by bacteria called group A streptococcus or the Streptococcus pyogenes. The gas gangrene is most commonly caused by infections with a bacteria called Clostridium perfringens. Bacteria gather in an injury or a surgical wound which has no blood supply. The bacterial infection starts to produce toxins that release gas and cause tissue death. And finally, the dysentery is an intestinal infection which is caused by a group of Shigella bacteria, which can be found in human gut. So our correct match would be A, E. coli matches with urinary tract infection, Streptococcus matches with sore throat, Shigella matches with bacillary dysentery, and Clostridium matches with gas gangrene. So our option number C is the correct answer. The next question is asking 0.1 molar solution of a weak acid has pH equals to 5 at a given temperature. What is the dissociation constant of that acid? So at equilibrium, we have HX equilibrium at H positive plus X negative ions. So since the pH is 5, for 0.1 molar, we have H ions at 10 to the power minus 5 molar plus 10 to the power minus 5 molars of X negative ions. Equilibrium constant of a weak acid is given by Ka H positive X negative upon Hx. So we'll just put the values here and we'll have the value of Ka, that is 10 to the power minus 5 into 10 to the power minus 5 upon 0.1 or 10 to the power minus 9. So the correct answer here would be option number A, 10 to the power minus 9. The next question is, which of the following is the principal buffer of intestinal fluid? The principal buffer in intestinal fluid is carbonic acid, which is formed by the combination of carbon dioxide with water and quickly dissociates into the bicarbonate ion. It neutralizes the acid that is released due to metabolism. So the correct answer here is option number A. Next question is, in monoclonal antibody production, the role of aminopterin in hat medium is 2. Hat medium is the one medium that is used to prepare monoclonal antibodies. Aminopterin in the hat medium blocks the de novo pathway. So typically, when the de novo pathway is blocked, cells will then utilize the salvage pathway or the alternative means to replicate. So here, option number C, blocks the de novo pathway of DNA synthesis is the correct answer. Our next question is, small molecule haptanes can be made immunogenic by. Haptanes cannot invoke an immune response by themselves. They can become immunogenic when they're joined to a larger or more complex molecule. This is a protein, a feature that is used in the study of immune response. So the correct answer to this question is option number D, conjugating the haptanes with larger molecular weight proteins. Our next question is, which of the following 5 major classes of antibodies can activate classical complement pathway? The activators of classical pathway include IgM and IgG. Classical pathway begins with the formation of antigen-antibody complex, or what we call as the immune complex. So when an antigen enters into the body, the antibody, which is either IgM or IgG, they bind to it. And they induce conformational change in the Fc portion of the antibody, which exposes a binding site for C1 proteins, the complement proteins. So the right answer to this question is option number C, says D, IgG, and E, IgM only. Our next question is, what is the correct answer to the following integral? So here we can see, this is the question that I've solved for you here. The integration of 1 minus sine x upon cos square x dx. So once we integrate it, you'll get 1 upon cos square x dx minus integration sine x upon cos x into 1 upon cos x dx. We can also write it as integration of sec square x dx and sec x dot 10x dx. And finally, after integration, you get this value 10x minus sec x plus c. That will be your option number A. The next question is, which of the following statements are true? First statement is, all bacteria have circular chromosomes. No, that's not true, because bacteria may have a circular or linear chromosome. That makes our first statement as incorrect and the second statement as correct statement. Next, yes, many bacteria have circular plasmids and the linear chromosome. So yes, they can have more than one chromosome. And finally, no bacterial chromosome is separated from the rest of the cytoplasm by nuclear membrane. So this particular statement is not correct. So the option number C, statement number B and C only is our correct answer to this question. Our next question is match the diseases in list 1 and the respective causal agents in list 2. The retrovirus, known as human immunovirus, causes acquired immunodeficiency syndrome or AIDS. Malaria is caused by protozoa of the genus Plasmodium. Although many of the viruses can cause common cold, but rhinoviruses are the most common cause. And finally, a common cause of bacterial pneumonia is Streptococcus pneumoniae. So the correct matches would be A goes with 4, retrovirus, malaria goes with protozoa, common cold goes with rhinovirus, and pneumonia goes with a bacteria, Streptococcus pneumoniae. So our option number C would be the correct answer. The next question is, it appears that signaling protein has a serine residue at position 96 that is phosphorylated upon activation of the signal. To confirm this, you plan to replace it by another amino acid so that it loses its function, a loss of function mutation. Which of the following amino acid would be your choice for its replacement? Now, you have to remember this. Alanine is the useful amino acid in any loss of function experiments, especially with respect to phosphorylation. In fact, some techniques involve creating a library of genes, each of which has a point mutation at different positions in the area of interest. And the simplest way to do it, and in fact, the first one which was used, is known as the alanine screening, where every position in turn is mutated to alanine. So correct answer here is option number B, alanine. My next question is, if the father has genotype IBI, capital IB and small I, and the mother has genotype capital IA and capital IB, then what proportion of progeny will have blood group O? Actually, if we look at these genotypes, there's no possibility that a child with O blood group would be born. So the correct answer to this question is option number A, 0%. The next question is, which one of the following compounds does not contain triple bonds in the given options? Now, here we have these given options, and if we look closely, ozone is the only one that does not have triple bonds. So our option number D is the correct answer. Next question is, which of the following expressions is correct? The universal gas constant is 8.3145, but not calories, it's joules. So the option A is incorrect. The Boltzmann constant is 1.3807 into 10 to the power minus 3 joules per K. Yes, this is correct. Planck's constant is 6.626 into 10 to the power minus 34. So no, this is incorrect. And Avogadro's number is 6.022 into 10 to the power 23 molecules per mole. So this is also incorrect, and our correct answer is option number B. The next question says, below are the metabolites that are formed in the TCA cycle. Choose the correct answer that indicates the order of formation in the TCA cycle. So here I put for you the TCA cycle, and first comes the ketobutyrate or the 2-oxobutyrate, then succinyl-CoA, then succinate, fumarate, and malate. So they're already arranged in the order. And now option number C, A, B, C, D, and E is the correct answer. Our next question is, match the description given in list 1 with the terms given in list 2. Okay, let's see. Statolysts are dense amyloplast organelles that synthesize and store starch involved in the perception of gravity by plants. Second, gravity sensing occurs in a few specialized cells which are known as your statocytes that are located in the center of the cap, an organ that covers the root apical meristem. In the shoot, the statocytes appear to be located in starch sheaths of the endodermal cell layers that surround the vasculature. The innermost layer of the plates is the starch sheath, which is gravity sensing tissue. It is specialized cell to store starch. They help in regulation of movement of ions, water, hormones into and out of the vascular system. And finally, the gravity sensing organ in their roots. So the gravity sensing organs are present in the roots. So let's see what is the correct match here. A matches with 1, B matches with 3, C matches with 2, and D matches with 4. And that's going to be your option number B, which is the correct answer. Next question is, mean free path of oxygen molecule is defined as where N upon V is molecules per unit volume, V is the molecular diameter, and lambda gives the mean free path. That's what we have to calculate. So in the kinetic theory of gas, the mean free path for particles, such as a molecule, is the average distance the particle travels between two successive collisions. And it is given by lambda is equal to total distance traveled by a gas between successive collisions upon total number of collisions. Now, during two successive collisions, a molecule of gas moves in a straight line with a constant velocity. And the mean free path of a gas molecule is given by lambda is equal to 1 upon under root 2 pi d square. D is going to be your molecule's diameter, N, which is your molecule, number of molecule per unit volume, that is N upon V. So from the given options, or option number A is going to be the correct answer. The next question is, which of the following organisms can be used as natural genetic engineer for transformation of japonica varieties of rice? We all know that Agrobacterium tumenifersens is a soil bacteria known as the natural genetic engineer, and it has a natural ability to genetically engineer a plant. So the correct answer to this question is going to be option number B. The next question is, arrange the following in decreasing order of amount of water absorbed or excreted in alimentary canal. We know intestine is the most important part of digestive system where much of the digestion and absorption of food takes place. As we know that parts of small intestine in descending order is duodenum, jejunum, ileum. The large intestine is also known as the colon. Water is absorbed here and the remaining waste is stored as feces, which is then removed by defecation. So the correct answer here would be jejunum, ileum, colon, and feces. That is our option number B. The next question is, which of the following strains of E. coli can function as donor of F factor? Now, before understanding the answer to this question, let us discuss that conjugation is merely the fusion of two compatible bacterial cells. Now, bringing two genotypes together and allowing them to conjugate is the equivalent of making a cross in eukaryotes. Now, conjugation and gene transfer in E. coli is driven by a circular DNA plasmid, which is known as the fertility factor or the F factor, which is found in some but not all cells. The cells that carry this F plasmid or the fertility factor are designated as F positive, and those that do not have it are known as the F negative. Now, in certain cells, F plasmid gets integrated into the host genome, and during F transfer, the host genome is also transferred. Therefore, such strains with an integrated F factor are termed as high frequency of recombination, or the HFR strains, to distinguish them from the normal F positive strain, which contain only a few rare HFR cells and thus display only low frequency of recombination for the strain as a whole. Now further, the integrated F factor occasionally leaves the chromosome of an HFR cell and moves back to the cytoplasm, in some rare cases carrying a few host chromosomal genes along with it. The modified F, called F prime, can transfer these specific host genes to a recipient cell in an infectious manner. Hence, F positive, HFR, and F prime, all of them can function as donors of F factor. So, a correct answer to this question is option number C. In the next question, which of the following statements is incorrect? The statement A is glycolysis involves conversion of a 6-carbon glucose to 2 molecules of 3 carbon pyruvates. That is true. Second statement is for each glucose molecule converted, 4 molecules of ATP is the yield of glycolysis. Actually, for each glucose molecule converted, we have 2 molecules of ATP as the net yield of glycolysis. So the statement 2 is incorrect. Statement C says, in eukaryotic cell, the citric acid cycle takes place in the cytosol. No, in eukaryotic cell, the citric acid cycle takes place in the mitochondria. So statement C is also incorrect. Statement D is both NADPH and NADH are electron donors and function in interchangeable manner. Rather, both NADH and NADPH serve as hydrogen electron donors for the reaction inside of the cell. NADH is mainly involved in catabolytic reaction, whereas NADPH is involved in anabolic reactions. The main difference between NADH and NADPH is the role of each type of the reduced coenzyme inside of the cell. NADH is used in cellular respiration. The NADPH is used in photosynthesis. So they do not function in an interchangeable manner. Therefore, statement D is also incorrect, and a correct answer is option number D, B, C, and D only. Our next question is, which one of the following combinations of statements regarding the extra embryonic membrane is correct? The first statement is correct. The amnion is the water sac that protects the embryo. Between the amnion and the embryo, there's an amniotic cavity, which is filled by amniotic fluid, which is secreted by both the embryo and the amnion. Amnion protects the embryo, while the amniotic fluid, it acts as a shock absorber and also prevents desiccation of embryo. Statement B is incorrect since chorion does not store the urinary waste. It is the outermost fold that surrounds the embryo in reptiles, birds, and protokaryons. And this particular allentochorion acts as an extra embryonic lung and helps in exchange of gases. In primates, however, including human beings, chorion forms the placenta. Further, statement C says chorion is a water sac that protects the embryo. Now that is also incorrect. The last statement is correct. Yolk sac is derived from sphingopurae, the inner endoderm and the outer mesoderm, that grows over the yolk to enclose it and helps mediate nutrition. The correct answer to this question is option number D, that is A and D only. The next question is, which of the following micronutrients, which of the following are micronutrients for plants? There are 7 essential plant nutrient elements that are defined as micronutrients: boron, zinc, manganese, iron, copper, molybdenum, and chlorine. These constitute in less than 1% of the dry weight of most of the plant. So from the given option, the correct answer is option number B, iron and chlorine. The next question is find the equation of a curve passing through point -2, 3. Given that the slope of the target to the curve at any point xy is 2x upon y squared. So the slope of the given curve is given by this particular formula dy upon dx, that is 2x upon x squared as it is given. So we can just cross multiply and write it like this. Then we're integrating both sides. We're integrating y squared dy and integrating 2x dx to get this particular thing x squared plus c. Now here you can see, oh, this will be x cubed upon y. Sorry, y cubed upon 3. When x squared minus 2 and y squared is 3, then we'll have 9 is equal to 4 plus c, and if we solve this, we get the value of constant as 5. So the equation of the required curve is going to be y cubed upon 3 is equal to x squared plus 5. I'm just putting in the values here. Sorry, let me just solve this particular thing. X cubed is equal to 3. So just cross multiplied it, 3x squared plus 15, or you can write y is equal to 3x squared plus 15, 1 upon 3. That is your option number A, which is the correct answer. The next question is arrange the following components in increasing order of acidity. And phenols, the presence of electron releasing group decreases their acidity, whereas presence of an electron withdrawing group would increase its acidity as compared to the phenol. Now, among the meta and para nitrophenol, the later is more acidic as the presence of NH2 at the para position stabilizes the phenoxide ion to a greater extent than when it is present on the meta position. So based on these facts, the correct order of acidity would be B, A, C, and D, and that is our option number C, which is the correct answer. Our next question is antigen pulsed peritoneal macrophages were taken from the mouse strain H2B, and antigen primed T cells from mouse strain H2K were added in the same well. T cell proliferation was then assessed. Now, since T cells are highly specific, the T cell proliferation would not occur until the antigen and the T cells are taken from the same mouse strain. So, the correct answer to this question is T cell proliferation would not occur. That is option number A. All right, then. Thank you. That's all for this video. I hope this was useful, and we'll catch you soon in our next video. Happy learning till then. Bye-bye.